Let's talk about how pH affects growth of microbes. So microorganisms are composed of proteins, just like eukaryotic organisms. And proteins, when they are in environments that are not beneficial or not optimal, the proteins can potentially change in shape and denature. So pH influences protein shape. So if the pH is optimal, the proteins work most effectively. But if pH changes, either increases, becomes more alkaline, or decreases, becomes more acidic, then the proteins will break down. They will denature. And so pH has a lot of influence on the growth of microbes. In our body, the majority of microorganisms grow best at close to a neutral pH. Think of our blood. Our blood has a pH of about 7.4, so close to neutral. There are organisms, acidophiles, that grow best in very acidic environments. And so microbes that are used in making buttermilk and yogurts, these are acidophiles. And we actually have one microbe that grows really well in acidic environments in our stomach, and that's Helicobacter pylori. And so, hold on, I thought I had a really cool picture of Helicobacter. Helicobacter pylori is a flagellated bacterial cell. It is a bacillus-shaped cell, and what it does is it grows in the acidic environment of the stomach, but it protects itself by producing carbon dioxide and ammonia around it so that the gastric juices don't damage it. And then it can actually burrow into our through the connective tissue, so through the epithelium, the connective tissue, and even the muscles surrounding the stomach, and that causes the ulcers that we hear about. And so treatment is often a proton inhibitor. So proton inhibitor is going to decrease the amount of acidic environment that's in the stomach, and then antibiotics that could potentially kill off the Helicobacter bacteria. Osmotic pressure is the pressure to pull fluids into the cell, back into the cell. So the amount of proteins inside of a cell will influence the ability to pull fluids back in. Hydrostatic pressure is the pressure that fluids have pushing out of a cell. And so the more fluid or water we have on one side of a cell, the less water on the other is going to influence water movement. And that's just basic osmosis. If you don't remember osmosis, definitely talk to me about it because I can explain this. And so we have organisms, barophiles, that can withstand very high hydrostatic pressures. So think about organisms living at the bottom of the ocean. These guys have so much pressure on them that they can't, that they have to be able to survive that environment. So they don't pull too much fluids into them, and they can survive. Halophilic organisms are salt tolerant, unless you're an extreme halophile. Then you might be a salt-loving organism. And they can maintain their own osmotic pressure by pushing salt out of the environment or pulling more salt in. And so Vibrio cholera, which causes the disease cholera, is an example of a halophilic organism. that can influence its own osmotic pressure. The last thing in this chapter that we're going to talk about are some different medias that we use. I've already talked about what media are. We have broths and we have our augers, but there are a lot of different types of augers and broths that we use. A complex medium is a medium that is undefined. And so the nutrient auger, tryptic soy auger, those are examples of complex media. Nutrient broth is a complex medium. Then we have some that are synthetic mediums that we know they're chemically defined. So we know exactly how much of every single element is in the media. In our lab, we don't use synthetic media. Those are used mainly in research labs where they have to grow bacteria under specific conditions so that their research works. We grow organisms on selective, or we grow organisms on complex media, but the media can have certain characteristics, like they can be selective or they can be differential, and so they grow only certain types of organisms so that we can identify the bacteria. Selective media are media that inhibit the growth of unwanted organisms. So it selects for what it wants to have grow. This allows us to only grow what we care about, and it doesn't grow other things. Phenyl ethyl alcohol agar, this is an example of a selective media. Oh, phenyl ethyl alcohol agar right here. This is a selective media that is selective for gram-positive bacteria, but specifically gram-positive coccus bacteria in the genus Streptococcus or Staphylococcus. The phenyl ethyl alcohol inhibits growth of gram-negatives, so gram-negatives don't tend to grow. So if you have a mixture of bacteria and you're trying to isolate gram-positives, you can actually use phenyl ethyl alcohol agar, and it might grow just what you want. So everything, so I want to make this very clear. If you are trying to isolate one type of bacterial species, that doesn't mean the other bacteria will not grow at all. If it inhibits growth, that means it's going to slow the growth down. It doesn't kill off the bacteria, so the bacteria may still grow, but they're going to grow at a much slower rate. We also have differential media. Differential media are media that contain extra substances that help us to separate organisms by how they ferment or how they do something. An example is blood agar. Blood agar is a differential media that allows bacteria to grow, but it differentiates them on how they break down blood. So Streptococcus pyogenes, or pyogenes, that's how I like to say it, is your harmful strep that causes strep throat. And this causes, or strep pyogenes, causes beta hemolysis of blood. So it basically breaks it down really bad. Alpha hemolysis of blood is caused by other streptococcal species like Enterococcus facialis. And so it doesn't cause disease the same way. And I believe there's also a gamma hemolysis, I think it's gamma, where there's no breakdown of blood cells at all. A lot of the media that we're going to play with are selective and differential. So they select for specific organisms to grow, and then if the organisms grow, they differentiate on the basis of if they ferment or if they utilize some resource. So they're selective and differential. Mannitol salt agar is a selective medium. It selects for the growth of bacteria in a relatively high salt concentration, so higher than normal. And then it is differential for bacteria that on the basis of bacteria that ferment mannitol, which is the sugar, and those that don't. So this is really important in identifying pathogenic and non-pathogenic Staphylococcus species. The pH indicator is phenol red, and so if the pH changes, phenol red will turn from a red to a yellowish color in an acidic environment. Pathogenic Staphylococcus aureus ferments the mannitol, and non-pathogenic species do not ferment the mannitol. So Staphylococcus epidermidis is a non-pathogen. Most of our gram-negatives and our gram-positives are not halotolerant, so they won't even grow in the environment. Vibrio fischeri is our only gram-negative that will grow in a salty environment, and it also ferments mannitol, so it will grow and ferment mannitol. Pretty interesting, right? Eosin methylene blue is selective and differential as well. It selects for gram-negative growth, so it does not allow gram-positive growth. Methylene blue and eosin are the inhibitory agents that inhibit gram-positive growth. And then it differentiates between those that ferment lactose and those that don't. So extreme lactose fermenters are going to typically produce this green metallic sheen. If you change the medium color, so right here you have growth, then you are positive for lactose fermentation. And if you grow but you didn't change color, then you are lactose negative. And all three are going to be gram-negative bacteria. I just said this. So lactose fermenter change color, extreme lactose fermenters turn that metallic green sheen. Typically we see this with E. coli, but sometimes E. coli doesn't produce that beautiful green sheen. The last thing we'll talk about is how we can measure growth then. So we might be working in a lab wanting to measure growth, or you might be getting samples from a certain environment, like a desert environment or a water environment, and they're asking you to tell us how much how many different bacterial species are here, or what is the total count of bacteria in the environment. And so we're looking at a sample, and then we have to infer the information to the entire environment. There are many different mechanisms to producing pure cultures. One is the pour plate method of isolation. It's a really, the idea behind it doesn't seem hard, but we've done this in our lab and it's really hard. So the method that we typically teach is the streak plate method of isolation. So if we're looking at, say, a sample of dirt from some pasture or something, they ask us to tell us, what is what type of microbes are here. We can take the dirt, we can put it into a broth, and then we can grow the broth and produce a streak plate that will allow us to have isolated colonies. Each colony here is going to have been started by one organism, one cell. And if you look, there are smaller colonies and larger colonies. So these could be different organisms. A lot of times you'll have different colors that will help you to identify the different numbers of organisms. We can count the number of cells using different mechanisms as well. So one mechanism is called the direct microscopic count, where we take a sample, we dilute the sample. So say we have initially, we have a sample from a lake, one of the lakes, Canton Lake, that would be an example. And we want to know how many bacteria are in the lake. We can dilute the sample to a small amount so that we now have less cells. And then we put the sample into a small counting chamber. And then we just look and we count the number of cells that we see. So this is beneficial in that it allows us to identify the number of cells in the sample, and then we take that information and we can figure out, okay, that was, in 1 milliliter of the water, and we diluted it to this amount. So the lake has, I don't know how much water's in the lake, but you could figure out approximately the amount of bacteria in the lake then. And that can be beneficial. But the cells that you're counting can be living, and they could also be dead cells. And so if we're not wanting to know, we just want to know the number of living cells, so dead cells aren't going to cause problems, then we'd have to use a different mechanism. Another mechanism is the standard plate count procedure. So the standard plate count procedure, we take a sample, again, like, a milliliter of fluid from some environment, and then we dilute it into a known amount of broth. We then dilute it further into more broth and more broth, so that we're diluting it like multiple times, taking 1 milliliter, or we start with a 10 milliliter tube of water. We take 1 milliliter out and put it into the first tube. We mix it up and we take 1 milliliter of that out, put it into another tube of 9 milliliters of broth, and we just keep diluting it further and further. Then we grow the bacteria, and only the living cells can produce colonies. So we will have a plate, we would assume on the lowest levels, where we've diluted it a lot. We would have plates that have individual colonies that we could then count, and then we could infer the information back to the lake or wherever we're getting our sample from. We can also use turbidity measures to determine how much waste is being produced, and if we know the amount of waste being produced, we can then determine the number of bacterial cells in the population. If you have a lot of bacteria, you're going to have a much more turbid solution. So the turbidity will be caused by the bacterial cells themselves, as well as the waste products that are produced. So that's it for this chapter. It's not a super long chapter. I hope you enjoyed it. Bring me questions, and I will see you in class. Bye-bye.